Oh, hi, Gareth. How are you?
Just as
How's
bored
life?
as you are.
Hi.
Stuck at home, no lab, nothing. But you look well.
Oh, thank you. Well,
Are you speaking first?
yes, I am going to,
Okay.
hopefully.
Are you in London or are you
Yeah,
in
yeah,
Spain?
yeah, yeah.
You collect butterflies?
Yeah, I like them for decoration purposes, let's say, yes.
Uh, yeah. Well, good luck with your talk.
Oh, thank you. Have you gone to the office at all lately, or?
I pop in every Friday since
Okay.
Christmas, mainly because they now do a free PCR-based COVID test,
Yeah.
which King's developed itself. It's not the government one, and it gives you the result. So Friday I do the test. Saturday they email and say I haven't got COVID. So
Good. Very
of
good.
course I can go to Borough Market and shop for nice food after that. So
Yeah. I
I
mean,
go.
people
I
get
go.
tested twice if they want in Queen Mary, no, Eva?
mm,
Twice a
Well, you
week.
can do it twice.
Yeah. Yeah.
I only do it once because apart from going out on the Friday, I do almost nothing. We have supermarket deliveries to home.
Mm.
We don't see our friends, neighbours, families, grandchildren, whatever. Just like everybody else. Just like everybody else. Nearly a whole year now.
Oh no.
I
I
think
mean,
it's
it's
really,
over, but it's getting better. So let's be positive
Yeah, yeah. And
and
then the sun shining.
optimistic.
And the
Yeah.
sun shining in London.
Yeah, the sun helps, I have to say.
Yeah. Okay, I leave you to your work.
Thanks, Gareth.
I think everyone's really reflecting on oh my gosh, it's March and thinking about where you were in March. And I keep remembering the conversation I had on February 28th when I was at a seminar, my last scientific in-person seminar that I've been to with a friend of mine. And I was just so excited for a few month lockdown because I thought it was going to be so productive. I knew it was
Yeah.
coming and it was, okay, I'm going to write this paper and I'm going to get all this done. And it'll be a couple months.
Yeah. Innocent we were a year ago. Ay, ay, ay.
All right, I think we'll just give it maybe like one more minute and then
Yeah, let's do another minute.
have you guys get started. Thank you, everybody, for continuing to show up each week. I know that we all have lots of Zoom fatigue, but it's great to see so many people here.
Okay, I think we can get started. We're on YouTube, right, Jan? I think we are.
Yeah,
okay,
we're there.
So let's get started. So hi, everyone. Just a reminder in case anyone's new, if you have any questions for the speakers, you can use the raise hand function, or you can type question in the chat, or you can just type out your question and we can read it out to the speakers after the talk. So we've got two speakers today. Our first speaker is Vicky Sanz-Moreno. Vicky started a group in 2011 with a career development fellowship at King's College London before joining Bart's Cancer Institute as a professor of cancer cell biology in 2018. She's been awarded the BSCB Women in Cell Biology Early Career Award Medal and received a CR UK Senior Fellowship. And a couple of years ago, the Distinguished Alumnus Award from Collegio Immaculada and Paper of the Year Award at Bart's. And today she's going to be talking to us about actomyosin cytoskeleton in cancer. Feel free to share your screen and take it away.
Well, thank you so much for having me here today. Can you see my screen? Yep. Okay. So I'm going to try and entertain you for 40 minutes or so and convince you that maybe the actomyosin cytoskeleton is quite an important structure in cancer, not only for migration, for a few more other processes, but that are linked to a migratory behavior. So to this audience, probably I don't have to explain too much how ROCK and myosin-2 control migration and invasion, but maybe just a reminder that the signals that this kind of cellular machinery are receiving are of different types. We have growth factors, chemokines, or cytokines that can activate RAW, and this activates it in a way that now it can lead to ROCK engagement, and either directly or indirectly phosphorylate myosin light chain, and together with the heavy chain generate actomyosin contractile force. And that leads to, of course, cytoskeletal rearrangements, but also transcriptional changes. And that means that the cell can change its behavior quite dramatically. So the other way in which we can activate the pathway can be through physical or geometric changes in the environment, let's say. And for example, mechanical tension is a way of activating ROCK that is not dependent on RAW and is very common in tumors. So once this pathway is active, we know that it plays a key role both for collective modes of migration or for individual migratory modes. In more elongated types of migration, we have the contractility is important for retracting the rear of the cell and at the protrusions. But then a hyperactivation of this pathway coupled to lower levels of adhesion leads to this more, let's say, I'm going to call it rounded amoeboid behavior in cancer cells. And the rest of my talk is going to be focused on this migratory behavior. So in the last few years, we've also proposed that because actin and myosin are required for literally all kinds of migration in tumors, this could be something that we should be thinking of targeting. If we want to block migratory plasticity, we could either use actin polymerization inhibitors or myosin inhibitors. And we've called these drugs migrostatics as opposed to cytostatics. But the truth is that maybe by altering the cytoskeleton of a cancer cell, we could also be altering their responses, not just cell migration. So in my lab, we're quite keen on understanding melanoma because this is a very migratory and invasive tumor that has to penetrate in the dermis, and it can, with quite a small lesion, can become quite invasive and metastasize early. So this is a behavior that is quite dangerous, and for that we studied on collagen matrices, collagen 1, which is the main component of the dermis, but also we studied because these cells are quite immunogenic, so it's a good model to understand the crosstalk within the immune system. And when you put melanoma cells on these collagen matrices, they can move using these amoeboid strategies. Let's see, I'm gonna try and play it from outside. And you can see these behaviors with blebs as functional protrusions, but also in 3D, we can measure their invasion, but also doing intravital imaging approaches that we've done in the past with Eric Sahai and Fernando Calvo. We can see that a lot of the behavior at the edge of the melanoma tumors, you see the purple is the collagen, is out of the total migration is either amoeboid or what we call amoeboid streaming migration, in which these are amoeboid cells kind of following each other. So in the last few years, we've been developing imaging pipelines for tissue, for human and mouse tissue, to really understand if specific pathways are active at the edge of these tumors, and what we call is the invasive front. And I hope you see that phosphomyosin, it's really high at the edge of the tumors. We've seen this in many of our studies, and this really corresponds with the cells when they are in contact with the matrix. Actually, this is mass staining, and you can see that some cells are still invading as groups, but there is also a lot of individual behavior. Cells are very rounded, and indeed they have very high levels of myosin when they're doing this. So we have been trying to generate a molecular fingerprint for these aggressive and invasive cells, just taking some omics approaches. And these studies are ongoing, but I'm going to kind of summarize a few of the latest things that we have observed. And Eva later will also tell you a little bit about their metabolic behavior. So we really are a signaling lab. So by doing these omics approaches, we understood a lot of the signals that these cells are receiving. But of course, there was no surprise here to see that Rho and CDC42 actually were very important to regulate actomyosin dynamics in these cells and drive the pro-migratory behavior. But what was maybe a bit more surprising through the omic studies was to see that not only your classical cytoskeletal regulators were very important for these cells, but signaling pathways that are protumorigenic were highly active in these cells, like STAT3 or NF-kappa B, which drive a pro-inflammatory tumor microenvironment. Surprisingly, they also secreted MMPs as a result of having high STAT3 and NF-kappa B signaling, and TGF-beta, which help their invasion. But also because of their secretory potential, and I will tell you a little bit about this, they are immunomodulatory, so they can affect the behavior of immune cells in the tumor microenvironment. What they don't like is reactive oxygen, that they like to keep it down. But also they are in a pro-survival program, and they crosstalk with a key oncogenic pathway in melanoma, which is MAP kinase signaling. And I will also go a little bit more in detail. And very recently, we also found that they have pro-cancer stem cell properties, and this is because they hijack some developmental programs such as non-canonical Wnt signaling. And melanoma is like a professional cancer in doing these behaviors, but we're starting to observe it in other epithelial tumors, not just, in epithelial tumors, because melanoma is non-epithelial, but we're not the only ones, of course. But we've published recently some behaviors in liver cancer, and then we have a lot of unpublished data in breast cancer or in pancreatic cancer. This is pancreatic amoeboid cancer cells, and these are invading into the matrix. They don't look that different from the melanoma cells. So these cells are at the edge of the tumor, so they could be communicating with the normal cells. And one way of communication is through secretion. So we have this model in the lab that we use quite a lot. These are very metastatic melanoma cells. They are 95% of them are amoeboid. They have very high levels of phosphomyosin. And we compare them to the less metastatic counterpart, which has less myosin, and we have a lower proportion of amoeboid cells, much lower. So we can see that when you have an enrichment in amoeboid cells, you become highly secretory, and these factors that these cells are secreting have the potential to affect other cells. Specifically, when we looked carefully, the prediction was that monocytes and macrophages should be affected in the tumor microenvironment. So Mirella, what she tried is first she knocked down contractility in any way you can imagine, and she did reduce this secretion. But what she wanted to do is, what was the functional consequence in the monocyte macrophage compartment? What she did is, to healthy monocytes, she gives some of this media, and I hope you can see that the monocytes became highly migratory, because this media was full of chemokines, by the way. And if you leave these cells in culture, they will differentiate into macrophages, and they will start expressing CD163, CD206. These are markers of pro-tumorigenic macrophages, as opposed to other macrophages that actually have tumor killing abilities. So we really tested this. We gave interferon gamma and LPS to monocytes. They differentiate into macrophages that actually have, I hope you can see, when we co-culture them with cancer cells, they kill them. In these co-culture assays, they have tumor killing abilities. But if the monocytes, the healthy monocytes, have seen media from amoeboid cancer cells, and now we culture those monocyte macrophages with cancer cells, now they are protected. They don't die. So you can imagine in a tumor, you're going to have different types of macrophages. And the more macrophages you have of this kind, the more protected the cancer cells are, and those tumors can grow better. So now bear with me, because I'm just going to show you correlation data, but it's kind of satisfying to see it in patient samples. So we always see this, that the cancer cells, they round up at the edge of the tumors. They become very rounded. They gain phosphomyosin. And this is really in the same areas where we can see the infiltration of CD163, CD206 positive macrophages. And I don't show you this, but it was also in proximity with vessels, with the vasculature. So can we perturb the cytoskeleton, and does this have an impact on the monocyte macrophage infiltration? So we took amoeboid mouse melanoma cells to grow them in a syngeneic context. These cells have a BRAF mutation, like 50% of melanoma patients. And when you inject them in the dermis in an orthotopic way in the skin, they start invading the matrix using, I hope you can see the cells are very round when they're invading, very, they have an actomyosin cortex. We can see it here in the tissues. So the increase in phosphomyosin is again evident in the mouse melanomas, and this correlates with the infiltration of the CD206 macrophages. Now, if we inhibit the contractility of the cancer cells, so we just treat with a ROCK inhibitor before, ex vivo, and then we never treat again so that we don't touch the tumor microenvironment, we don't affect any other cell. The interesting thing is, even if these cancer cells have not seen any treatment for 14 days, we have significantly affected their invasion. They are not getting into the dermis. They are losing the rounded behavior and the myosin activity levels, the phosphomyosin. And now we can also see that the general effect in macrophages is that we no longer have this infiltration of CD206 positive macrophages. And I don't show you this, but also we had a big defect in tumor growth. So mechanistically, we took that secretome and we could see that it was heavily connected to NF-kappa B. And only when you knock down NF-kappa B in the cancer cells and we give this media to monocytes, we can see a defect in the recruitment of the monocytes, because when we try and knocking down individual chemokines, we were unsuccessful because there was too many of them. Then when we knock down again NF-kappa B or myosin in the cancer cells, and then we take that media and we leave the monocytes to differentiate to macrophages, we can see that this media now has lost a lot of the factors that made them macrophages of the CD163, CD206 kind. And we could rescue these with interleukin-4, which is one of the factors that they were secreting. That's not the only cell they are affecting. We could see from the media that they were secreting that there was a potential to affect endothelium. So what we did is to take the media and give it to an endothelial cell monolayer. And I hope you can see that they are making holes in the media because it was full of permeability factors. So this permeability could be rescued if, again, we knock down ROCK or NF-kappa B in the cancer cells, and then we took the media and gave it to the endothelial cells. You see now how the V-cadherin junction index is recovered. And this is because actually we could see via phosphoproteomics that the endothelial cells, when they see the conditioned media from amoeboid cells, their own contractility and their own cytoskeleton is very affected. So we could see both in vitro and in vivo that this media cause permeability in the endothelial cells and here in the lung vasculature. So when we knock down ROCK or NF-kappa B, you see that now the contractility is reduced in the cancer cells. Now we take that media, we give it to endothelial cells, and permeability is reduced. And we see exactly the same in vivo in the lung. And this results in a reduction of the lung metastatic colonization of these cancer cells. So this could be one of the mechanisms these cells use to colonize the lung. They are hijacking mechanisms that immune cells can use to, let's say, circulate into different tissues. So just a summary of some of Mirella's observations. When cancer cells reach the edge of the tumor, they gain myosin activity. And this leads to the secretion. I didn't show you this, but a particular molecule, interleukin-1 alpha, activates NF-kappa B. And now these cells can express and produce lots of factors that have an impact on monocyte recruitment, macrophage differentiation. These macrophages now help the cancer cells grow. And I didn't show you this, but they also help the endothelial cells proliferate. And once the cancer cells reach the secondary site, for example, the lung, they open up the vasculature. And that's how we think part of the mechanism, why they're so good at colonizing a distant site like the lung. So this was quite nice biology, but Jose in my lab was quite interested in the more translational aspects. How could we help melanoma patients with our studies? So for those of you who don't know this, melanoma is poster child for both targeted therapies directed to the key oncogenes, The key oncogenes are NRAS and BRAF that are mutated, leading to a MAP kinase activation. So this has led to the development of BRAF inhibitors and MEK inhibitors. And on the other hand, very promising approaches in the field of immunotherapy. We know that CTLA4 and PD-1 and PD ligand inhibit T cell function, cytotoxic T cells. So there is antibodies given to patients, anti-CTLA4, anti-PD-1 or anti-PD ligand. And this again is improving the outlook of melanoma patients. But unfortunately, many patients don't respond or develop resistance. So we thought these couple of studies were quite interesting because they describe a type of resistance that was not dependent on the acquisition of new mutations. On the other hand, what they describe is that melanoma patients that do not respond to MAP kinase inhibitors, that they acquire resistance, they upregulate a set of genes that not only sustain proliferation, but are very important for invasion and metastasis, but also for immune evasion. So this would mean that cells that are very invasive and metastatic are also very good at evading immune attack. Importantly, they saw that also a year later, the transcriptomes of the patients that did not respond to immunotherapy had an upregulation also of genes that are related to tumor invasion and ECM remodeling and so on. So we thought, could it be that ROCK and myosin, by virtue of regulating both invasion and immunosuppression, and specifically the crosstalk with macrophages, could play a role in the acquisition of resistance? So first of all, Jose, in an unbiased manner and in collaboration with Amin Sadok and Chris Tape in London, they did phosphoproteomics in amoeboid melanoma cells with a BRAF mutation, and they put a MAP kinase inhibitor, and very quickly they could see that the phosphoproteome was changing and many pathways were changing, very interesting ones. But the one that had the highest change was cytoskeletal remodeling and ROGDPA signaling. Of course, we have also here EMT and FKB, things that were also interesting to us. But first of all, what Jose looked is at the cytoskeleton of these cancer cells when they were treated first with a ROCK inhibitor that we know will have an impact on ROGDPA's outputs, but also in the cytoskeleton. You can see how actin and myosin no longer have the same configuration. We lose this amoeboid phenotype. The cells are pretty much collapsed. We can quantify it here, but interestingly, also the drugs that are given to the patients, the BRAF inhibitors or the MEK inhibitors, did something similar to the cytoskeleton. So we can place ROCK and myosin downstream of this oncogenic signaling, and this makes sense that an oncogene would use some cytoskeletal related pathway, not only to transform the cells, but also to make them more invasive, but also more resistant to the therapy. So when we look at the timescales in which things are changing, we know that long term, if you leave these cells with a BRAF inhibitor, eventually after months, they will recover MAP kinase activity, and that's how they also become resistant. So we just left them for 48 hours. I hope you can see here that phospho-ERK is down. This is what the drug is trying to lower MAP kinase activity. And it's starting to recover, but very, very mildly. But I hope you can see that myosin activity is recovering very fast, very, very fast. And we hypothesize that the reason why now myosin is recovering is to provide a pro-survival signal in the presence of the drug. If the cells want to become resistant, they need to survive in some other way. And to kind of test this hypothesis, Jose overexpressed a mutant version of myosin that looks like if it's always phosphorylated by ROCK. I hope you can see that this myosin goes to the cortex. This is in 2D, but bear with me. And this myosin, indeed, when it's overexpressed and we see the drug, the BRAF inhibitor, it gives a pro-survival advantage to these cells that now are overexpressing this myosin. So when we look carefully at these cytoskeletons in sensitive cells, you can see very nicely actin and myosin together. There is some nice stress fibers. But when we put the BRAF inhibitor in the sensitive cells, we lose all that organization. Now, the resistant cells that have been generating over a few months of seeing the drug, they no longer are responding to the drug. You can see it here. MAP kinase is recovered, but also myosin is recovered, phosphomyosin. and the cytoskeletal structure. And this we saw in many, many models. Jose did a lot of work. You can see here that BRAF inhibitor decreases myosin in sensitive cells, but in resistant cells that have recovered, ERK myosin is recovered. And I don't show you this, but now these cells don't die with a BRAF inhibitor. They should die because they have a BRAF mutation, but they don't. So let's understand if maybe now this is a vulnerability of the resistant cells. You see here in this particular model, a BRAF inhibitor decreases myosin, but in the resistant cells, myosin is up. And now this myosin activity is dependent on ROCK because the ROCK inhibitor reduces myosin. And importantly, with the BRAF inhibitor, these cancer cells are very happy and alive, but they die with a myosin inhibitor, or here I show you with a ROCK inhibitor, we can use a very, very low concentration of ROCK inhibitor to kill these cells in comparison to the cells that die with a BRAF inhibitor. They are more resistant to a ROCK inhibitor. So we have a switch in their sensitivity. It's like a vulnerability. Now they're vulnerable to myosin and ROCK inhibition. And I tell you, Jose did this in a number of patient-derived models. He used different ROCK inhibitors, different RNAs against either ROCKs or myosins, and he always got the same result. And when we looked at the transcriptomes, what we can see is that our amoeboid cells that have very high myosin have a transcriptome very similar to the patients that are resistant to either MAP kinase or to immunotherapy. And those genes have to do with epithelial to mesenchymal transition, angiogenesis, hypoxia, wound healing, TGF-beta, and then STAT3, NF-kappa B that are very familiar to us. These are all cells that the amoeboid cells really like, and also mechanosensor JAP. So we did look into patients to see if we could see this phenomena in real patient samples in which we, in all these patients, did not respond to the therapy they were given, either MAP kinase inhibitors or immunotherapy or both. And I hope you can see that before and after receiving this treatment, phosphomyosin is very much upregulated, and this coincides with an increase, a massive increase in collagen deposition. So this is just a correlation, but we speculate that maybe all this collagen deposition could increase the contractility. But what we can see for sure is an immunosuppressive microenvironment. Again, we see the CD206 positive macrophages are upregulated, and a new immunosuppressive population has been also identified in these infiltrates. We see the regulatory T cells, FOXP3 positive cells, which we know dampen the immune response. So we have something happening in the cancer and something happening in the microenvironment. So if we look just at what happens in the cancer, why would they die in the presence of a ROCK inhibitor when they are resistant to their MAP kinase therapy? So we look at the transcriptomes, and these resistant cells have a massive deregulation of a reactive oxygen species metabolism and a downregulation of a DNA damage response. Therefore, we knew from our previous studies that if you have very high myosin, you like to keep your ROS levels low. So in the sensitive cells, we put a ROCK inhibitor and we can increase reactive oxygen to a moderate level that is compatible with some DNA damage, but they can more or less cope with it. But the resistant cells now have much more reactive oxygen inside them when we've put a ROCK inhibitor, and it's translated in much more DNA damage. So Jose did a lot of cell cycle analysis. The cells had lots of defects by fax, but importantly, when we follow them via video labs, we could see that they were starting to generate a phenomenon of multinucleation, and all these multinucleated cells couldn't cope with all these defects and died. And that's how we can kill them better. So if we take this to a preclinical model, and now bear with me because I'm going to, the things I'm going to explain, we're using systemic treatments, which is what the patients would see in the clinic. So now we're treating everything, tumor cells and stroma. But this is the model of resistance in which Jose applied the BRAF inhibitor, but also moderate, or let's say a low dose of ROCK inhibitor, because we think they are more sensitive to this, and also we can keep the toxicities low. And I hope you can see that even with this low dose, the combination therapy reduced the tumor volumes and reduced the myosin activity within the tumors. We cannot completely kill it, but we reduce it to some extent. And also, we are able to decrease the infiltration of the CD206 positive macrophages. If we go now to the immunotherapy scenario, we need a syngeneic model again. And it's difficult to model resistance, but Jose managed to do it. He has this melanoma mouse cells grown with a fully functional immune system in the mice. He treats with anti-PD-1, and what he gets is very variable responses. Some tumors respond and regress completely, and some tumors don't respond at all. And so he took the non-responders and regrafted them into a new mouse, and then treated a new mice, and then treated again with anti-PD-1 to sustain the resistance, and then do a combination therapy. So in this super resistant scenario, I hope you can see again the combination therapy has best results. The tumors are regressing and the mice are doing better. So let's look at the cancer cells. The ROCK inhibitor actually is working, is reducing the contractility. I hope you can see that the cells are very spindle now. But on top of that, we know that one of the ways in which cancer cells fool the immune system is using this PD ligand, because that impairs T cell function. So PD ligand was high in the cancer cells, but it was reduced with the ROCK inhibitor. And the cancer cells were not the only immunosuppressive, let's say, populations there, because the CD206 positive macrophages in this model of resistance were very high, and they had a very high score of PD ligand. But with the ROCK inhibitor, we can reduce it. And when we look at T cell infiltration, regulatory T cells were very much reduced with the ROCK inhibitor. And we think this is because ROCK regulates TGF-beta, and TGF-beta is a FOXP3 inducer. So we can see both in the mouse lines and in lines we had for immunotherapy resistant patients that TGF-beta is very reduced. On the other hand, it was important to see that we were not affecting cytotoxic T cell infiltration on other populations. And I hope you can see that these are infiltrating the tumor at the same levels as the control. So Jose concluded that the therapy that we are giving is changing how cells are responding. A lot of cells are dying, but the ones that are resisting are changing their cytoskeleton, and is providing a dual action, an intrinsic pro-survival signal, because now with high ROCK, we are protecting from reactive oxygen, but also an extrinsic signal to other cells that are infiltrating the tumor, like regulatory T cells and macrophages that are actually helping the tumor to grow. So with the ROCK inhibitor, we can get rid of this cytoskeletal remodeling, we can get rid of these immunosuppressive populations, and also cause some intrinsic cell death. So this is starting to point to us like amoeboid melanoma cells could have cancer stem cell properties, because they are invasive and they have drug resistant properties. And this is what in epithelial tumors has been associated with EMT and stemness. So we looked carefully at the transcriptomes again, and here Irene, what she could find is this very interesting set of enrichments, which is that the amoeboid cells have very high expression of neural crest stem cell genes, and cancer stem cells, and other types of stem cells. And then I just remind you that melanoma derives from melanocytes, and melanocytes are also their own, they come from the neural crest, which is a very highly migratory and multipotent population. So many of these genes actually we think are serving the melanoma very well for their purposes. And when we treat it with ROCK inhibitors, many of these genes are downregulated. So we took very spindle melanoma lines, Irene took them, of course, and then did serial melanosphere passaging, which what it does, it enriches in cancer stem cells and the spheres get bigger and bigger. So you can see here how they get bigger and bigger in these two models of spindle melanoma. And now when we look at the contractility of the spheroids, now the spheroids have more and more myosin activity. And to our surprise, also, they are very proliferative. They're proliferating more and more with the passages. So now we disgregated the spheres and we put the cells on collagen. And I hope you can see how from passage 0, where they form very small spheres, to passage 3, where they make big spheres, we see that the cells have gained roundness, In the two models, they've gained phosphomyosin. It's very clear that they're gaining this actomyosin cortex, and they are starting to bleb, which is a hallmark of high contractility in these cells. And when we looked at melanomas invading, again, we have injected intradermally mouse melanoma cells with BRAF mutations. We can see that in the distal invasive front, they are very far away in the dermis. They're not only very high phosphomyosin levels, they are also Ki67 positive, so they are proliferating. So we think these cells are very dangerous because they are leaving the tumor with the ability to keep proliferating. They're not switching off proliferation. So again, cancer stemness has been linked to EMT, and already with Jose's data, we could hint that maybe amoeboid cells express EMT genes, even though this is not a canonical EMT because these are not epithelial tumors. Let's call it a pseudo EMT or EMT-like process or gene signature. But these amoeboid cells have very high expression of all the genes that have been related to EMT and metastasis. And they are downregulated with either ROCK inhibitors or myosin inhibitors. But I can tell that this was a 24-hour treatment, so this could be an adaptation of the cells to the drug. So what Irene did here is a more kind of directed approach, 4 hours of ROCK inhibitor and a qPCR array directed to only EMT genes. So this is very quantitative, and I hope you can see that it doesn't matter the oncogene that is driving melanoma, if it's BRAF or NRAS, in amoeboid cells. When you put a ROCK inhibitor, there is an astonishing downregulation of EMT-related genes. So can these genes regulate amoeboid behavior in turn? So we took the genes that had never been linked to amoeboidness before, and we did a series of screens that we do regularly in the lab to test for amoeboid behavior. As a positive control, we use SERPINE1 because it was in our EMT array regulated by ROCK and has been linked to amoeboid behavior. And I hope you can see with SERPINE1, we lose the roundness when we knock it down. We lose myosin activity. We can now reattach the cells to the keratinocytes because normally melanoma cells, when they invade, they lose the attachment to the surrounding keratinocytes. And in a 3D invasion assay, also SERPINE1 decreases invasion. So now the two genes that actually had the very same effects in all assays were non-canonical Wnt ligands. So that was quite interesting for us because we know also that these are very important molecules for neural crest migration itself. So what Irene did, she did a lot of experiments to prove that these ligands drive the amoeboid behavior and their stemness. She also did the same with the receptors because they had very high expression of the receptors. But just to cut a long story short, I'll tell you what she did to elucidate if either this branch of the pathway, the TAM1 or the PLCB branch of the pathway, were controlling this phenomena. So when you give Wnt11 to very spindle cells, they become more rounded. If you remove PLCB from the cells, they can still round up to some extent. But if you remove DAM1, they remain completely spindle. And indeed, DAM1 knockdown was the only one having an effect on melanosphere formation and 3D invasion. So when we give Wnt11 to the cells, you get a very high raw activation that translates in high levels of myosin activity. But if you remove DAM1, this is no longer observed. So we remove DAM1 permanently from cancer cells, and then we try and see their potential to initiate tumors. And I hope you can see that now, without DAM1, the melanoma cells have a much harder time, a tumor initiating frequency that is much lower. You need many more cells to form a tumor. And this translates in a reduction of tumor growth when you have limiting concentrations of cancer cells, not when you have a lot of them. And when we look at the tumors, what we can see is we've lost the amoeboid invasive compartment. You can see it here, they're very round in the control, but with DAM1 knockout, they become spindle. The contractility at the edge of the tumor is very reduced, and so is the proliferation. So we wanted to, again, even though this is correlative information, but it tells us if in patients we can find markers of amoeboid cells, it means that those patients maybe would benefit from therapies against amoeboid behavior. So we see again in this large cohort of melanoma patients, the cell rounding, the myosin activation, but also that the cells at the edge of the tumor are not only invading, they are proliferating. And indeed, they express DAM1 They have very high levels of DAM1, and they are seeing the ligands. There's high levels in those areas of non-canonical Wnt ligands. And also cancer stem cell markers, which again makes them very dangerous, because what Irene saw is that the cancer stem cell potential of these cells is recapitulated in the metastasis. I don't show you the data, but she saw the exact same behavior. So we have found how these melanoma cells hijack a very important pathway for development, but now they use it, because these ligands are in the tumor microenvironment, they use it to activate invasion, proliferation, and starting a new tumor, both in the primary and the secondary site. So I want to even complicate your life a little bit more, just to give you the idea that not only myosin has a massive impact on the tumor microenvironment. In collaboration with Kevs Hodi-Valarilke at Barts, what we've been able to observe is that the tumor microenvironment itself, in this case, the mural cells, the cells around the endothelial cells, when they lose adhesion, beta-3 integrin, they activate NF-kappa B. And now this turns on some chemokines and cytokines that activate MAP kinase, ROCK, and myosin in the tumors. And therefore, these tumors are now growing more. So we need to cut the communication, all this secretory rewiring between the cancer cells and the tumor stroma. So I just want to leave you with this idea that we believe, because these cells are, I hope you can see here, they are at the edge of tumors. This is phosphomyosin. They are the cells that are going to leave and access the vasculature. And they have all these horrible properties. Not only they're invasive, they have cancer stem cell properties, drug resistance, they are immunosuppressive. So the surgeons for melanoma already leave a very wide margin when they remove the tumor, which we totally agree, because those are the cells that we want to get rid of. And what we propose is not only we need to leave that wide margin, we should give therapies against amoeboid cells. And we have discovered a series of pathways that we think are actionable in the clinic, like JAK inhibitors, NF-kappa B inhibitors, or hopefully one day there would be good non-canonical Wnt inhibitors. So we do think we need to get rid of these cells. So with this, I want to thank you all for listening. I want to thank the whole lab, present and past members, our amazing collaborators, and all our funders who we couldn't do anything without. Thank you.
Thanks, Vicky. That was super interesting. It's nice to see I haven't seen a more translational talk in a while, so it's good to see how these things could go into therapy. So we got a question from Juanma in the chat saying, how are these transforming factors passed on the medium? Are they from multivesicular bodies or from shed blebs? Blebs have been seen to break from cells in vivo and harbor signaling factors from the environment. And I wondered if you saw the same, since the secretions are rock dependent.
Very good question. So in Mirella's work, we focused on classical secretion that we could inhibit with Brefeldin. And most of these are soluble cytokines and so on. But now we have unpublished data to show that also they're very good at making vesicles. And now we are, as we speak, trying to do some proteomics to see what's in those vesicles, if it is some of these factors or maybe others as well.
Great, thank you. That was a really enormous amount of data, really incredible work. So no, it's I'm so impressed. I'm like, it's really great. So our next question is from Anna Pasapera. She asks, did you look if the MAP kinase activation contributes to MLC phosphorylation through ROCK, myosin phosphatase, MYPT1 inactivation? And did you look at phospho MYPT1 inactivation sites?
So I think you're going to hear a lot about MYPT in the next talk, so I'm not going to anticipate anything. But what we have not found yet is the connection with MAP kinase, but this is something that Jose Orgaz, who has now his own lab, is going to look at in detail, so stay tuned. But I leave the MYPT connection for Eva, because she has a lot to say.
Okay, great, thank you.
Ismail Ortiz asks, what do you believe is the advantage of cells using amoeboid over mesenchymal migration?
Well, I hope I have shown you that it's not just about migration, because if you're very migratory, but then a T cell comes and kills you, or a macrophage, there's no point to that. So I think what they are very good at is at coping with all kinds of stresses. Through, cytoskeletal remodeling, they have adapted to different circumstances in which they overperform the mesenchymal cells, at least in our hands and at least in melanoma. I'm pretty sure there will be other things in which mesenchymal cells are better at. Yeah.
And to follow up on that, then do you really think of it just as sort of an evolutionary pressure, sort of selecting for the cells that happen to be able to have this sort of multifaceted ability to really thrive?
Yeah, so because we see that some oncogenic signaling sustains it very strongly. So this can be very hardwired and acquired over time. But we also know that if you give a fast cytokine or chemokine stimuli, and many other wonderful scientists have seen that, for example, confinement induces this very quickly. So we think there is two types of regulation. One that is just a short-term thing that happens and the cell can adapt. But if you give this signal for a very long period of time, and we see it, we have lots of melanoma cells that retain these features in culture without giving them any stress. They are just amoeboid. They have that in their memory of their cytoskeleton. So
okay, that's great.
Both.
Yeah, it really complicates sort of the overly simplistic view, I think, that I had in my head of sort of, okay, well, the cells will migrate out in this amoeboid, or sorry, in this mesenchymal sort of morphology, and then it turns out that it's right.
There's so many.
I know, couldn't you just keep it simple for me? So I think we're actually just in the interest of time, we're gonna move on to Ava now. So is it Eva or Ava? Sorry.
Eva.
Ava, perfect. Okay, thank you so much. So thank you, Vicky, for that wonderful talk. And now we are going to hear from Eva Crosas Molist. So she received her PhD in the lab of Isabel Fabregat, where she studied the role of the NADPH oxidase NOX4 in the pathophysiology of the liver. She's now working as a postdoc in the lab of Vicky, Professor Vicky Sanz-Moreno, who obviously we just heard from. So she's now supported by a fellowship from the Fundación Ramón Areces, and she's now studying research on the crosstalk between the cytoskeleton and mitochondria during tumor progression and invasion. So I think that's what we will hear about now. So thank you so much. Ava, take it away.
Thank you for the nice presentation. I hope you can see my screen.
Yeah, it looks great.
Okay, perfect. So today I'm going to show you a work in which we try to show that AMPK is a mechanometabolic sensor linking mitochondrial dynamics to myosin 2 dependent cell migration. So, as I told you, Vicky, before, cancer cells can migrate either as collective groups or as individual cells. And within the individual cell migration, the two main modes of movement are this elongated mesenchymal mode of movement or the rounded amoeboid migration, which they are interchangeable both in vitro and in vivo, which is what we know as cellular plasticity. And among other characteristics, these two types of movement, they differ in their cell morphology, in their myosin 2 activity, and in their adhesion to the matrix. On the other hand, we know that cells require energy to migrate. However, the connection between the cytoskeleton and metabolism is still poorly understood. So how the metabolism is required to fulfill energy demands during 3D migration is not very known. So we hypothesize that cells adopting different types of migration may exert different attraction forces on the matrix, and this may imply that they have different energy requirements. So to study that, we developed a method to measure traction stress in cells cultured in 3D by directly tracking the collagen fibers around the cells. So very briefly, we embedded live ACT expressing cells in a collagen 1 matrix, and we took stack images over time of the cells and of the matrix. And from these movies, we integrated the information of the matrix displacement directional vectors and depth information that, together with the stiffness of the matrix, allowed us to measure the traction stress. So we applied this method to two different cell types, HT1080 fibrosarcoma cells as a model of elongated mesenchymal cells, and A375M2 melanoma cells as a model of rounded amoeboid cells. And as you can see here, elongated mesenchymal cells induce much stronger stresses of much higher magnitude compared to rounded amoeboid cells. But interestingly, both the stress were myosin 2 dependent, since when we treated the cells with blebistatin, we were able to abolish both. Since measuring traction stresses is a tedious process, we defined intrinsic patterns of the different modes of migration in a panel of cell lines. This panel of cell lines include HT1080 fibrosarcoma cells, MDA-MB231 cells, and 4 melanoma cell lines. These melanoma cell lines are grouped in pairs. So there is this WM983A and WM983B, that they come from the same patient, the A from a primary tumor and the B from a metastatic tumor. And then we have A375 parental and A375M2, that they also share the same genetic background, but they have different metastatic potential. They also differ in their conductivities. So as you can see here, cell morphology is closely linked to the myosin 2 activity. And we found that these 4 cell lines adopt this elongated mesenchymal mode of movement. So they are the most elongated, showing a very low roundness index, and they show also low levels of myosin 2 activity, while the WM983B and A375M2 cells adopted this rounded amoeboid mode of movement, and they show a high roundness index and high myosin 2 activity. But the different modes of movement not only differ in their myosin activity, but also in the localization of this myosin. And as you can see here, in rounded amoeboid cells, myosin forms this contractile ring at the cortex of the cells. Different modes of migration also differ in their adhesion to the matrix. And so we analyze here and the points of attachment of the cells to the matrix, and we have found that elongated mesenchymal cells have many more points of attachment to the matrix compared to the rounded amoeboid cells. And this is in correlation with their speeds of adhesion to the collagen matrix. So I've shown you that cell morphology, cortical myosin 2 activity, and cell to matrix adhesion define the mode of migration of the cells. And this is connected to the magnitude of the traction stress that they exert on the matrix. But how is this? Is this connected to the energy requirements of these modes of migration? So this is what we studied next. And for that, we did an NMR metabolomics, comparing the metabolome of the 2 pairs of melanoma cell lines that I introduced you before. And we found that their metabolomes were statistically separable. Also, we found that the amoeboid cells were enriched in a particular set of metabolites. Cancer cells robustly engage in aerobic glycolysis, what is known as Warburg effect, to obtain energy in a very fast way, but also engage in mitochondrial metabolism to obtain building blocks and ATP for cell proliferation. We analyzed using a seahorse analyzer. We couldn't find any correlation between the mode of migration and their engagement in glycolysis, but when we analyzed mitochondrial respiration, we found a very nice correlation between the oxygen consumption rate of the cells and their mode of migration. This was much higher in elongated mesenchymal cells than compared to rounded amoeboid cells. And also these elongated cells produce much more ATP linked to mitochondrial respiration. And we were able to confirm this in 3D, where also elongated cells showed higher mitochondrial respiration. Also, the ATP levels, we could corroborate them in 3D using this Perceval HR fluorescence biosensor, in which we found, that HT1080 fibrosarcoma cells had much higher ATP-ADP ratio compared to rounded amoeboid A375M2 cells. Also, we found that rounded amoeboid cells had much higher levels of AMP compared to their elongated counterparts, and this will become relevant later in my talk. So given that the main differences we found were in mitochondrial respiration, we decided to focus our attention in mitochondria. And we analyzed the mitochondrial morphology in our panel of cell lines, and we found that in rounded amoeboid cells, mitochondria were spherical and fragmented, while mitochondria of mesenchymal, elongated mesenchymal cells were long and formed a network. I think you will see this very well in these movies of H75 parental cells and M2 cells. In red, you can see the mitochondria here very long, and in amoeboid cells in a dot shape. And also, we found that mitochondria were mainly located near bleeding regions, which are likely to represent regions of high energetic demand. So here I show you the quantification of the mitochondrial circularity. And what's interesting is that it's not only the morphology of the cells what is changing, but also their activity. So elongated mitochondria from mesenchymal cells are much more active than fragmented mitochondria from rounded amoeboid cells. So cells regulate the mitochondrial morphology through mitochondrial dynamics, which in turn is regulated from mitochondrial fusion and fission proteins. So we have mitofusin 1 and 2 that mediate the fusion of the outer mitochondrial membrane, and OPA1 that mediates the fusion of the inner mitochondrial membrane. And in agreement with their mitochondrial morphology, we found that rounded amoeboid cells had lower levels of mitochondrial fusion-related proteins compared to their elongated counterparts. To further understand the relevance of mitochondrial dynamics in the cell migration plasticity, we knocked down mitofusin 1 and 2 in elongated mesenchymal cells, and this not only changed the mitochondrial structure towards fragmented mitochondria, but also induced a transition to a more rounded cells to membrane bleeding, and it increased the myosin 2 activity in these cells. Here you can see the quantification of what I shown you, and this switching phenotype resulted in an increase in the invasive capacity of these cells in 3D, and it decreased their adhesion to the extracellular matrix. We found very similar results when we knocked down OPA1 that I told you that it mediates the fusion of the inner mitochondrial membrane. So what I've shown you so far is that through mitochondrial dynamics, we are able to modify the cytoskeletal remodeling. But how mitochondrial dynamics is linked to cytoskeleton was still missing. So what we know is that myosin is mainly regulated by the phosphorylation of its myosin light chain, and this can happen either directly through kinases or through the modification of the activity of the myosin phosphatase. And we have here MYPT1, that is the regulatory subunit of this myosin phosphatase, and when it gets phosphorylated, it gets inactivated. Therefore, it leads to a higher levels of myosin 2 activity. In a chemical genetic screen, MYPT1 was identified as a direct substrate of AMPK, which is a metabolic sensor. AMPK gets activated when there is an imbalance between ATP, AMP, and ADP. And I showed you before that in rounded amoeboid cells, we have low levels of ATP and high levels of AMP, suggesting that they may have an overactivation of the AMPK pathway. So we hypothesized that maybe mitochondrial dynamics was regulating the cytoskeleton via AMPK and MYPT. So we decided to study that, and we first compare rounded amoeboid cells versus their elongated counterparts, and we found indeed an increase in the phosphorylation levels of AMPK that were accompanied by an increase in the phosphorylation levels and therefore inactivation of MYPT1. And this would lead to a higher myosin activity that I told you that these rounded amoeboid cells had. Moreover, when we inhibited AMPK using an AMPK inhibitor in rounded amoeboid cells, we found that we also decreased the phosphorylation levels of MYPT1, and these cells lost their amoeboid features. Conversely, when we activated AMPK using an AMPK activator in elongated mesenchymal cells, we also increased the levels of phosphorylated MYPT1, and we induced a transition towards an amoeboid behavior in these cells. And strikingly, when we silenced Mitochondria 1 and 2 in elongated cells, we found also an increase in AMPK that was accompanied by an increase in the phosphorylation of MYPT1, and this transition towards this amoeboid phenotype that was prevented by compound C, the AMPK inhibitor. So I've shown you so far that upon mitochondrial fusion-related proteins loss, there is mitochondrial fragmentation that leads to an increase in phosphorylated AMPK, which inactivates myosin phosphatase, leading to an increase in the myosin 2 activity that increases the contractility of the cells, favoring this rounded amoeboid behavior. But I told you at the beginning that the different modes of migration are also differentiated by their adhesion to the matrix. So we wondered whether adhesion was also playing a role in this mechanism, because I told you at the beginning as well that we thought that the traction forces that these cells, the cells adopting different modes of migration exert on the matrix, which is intimately linked with adhesion, might regulate the energy requirements of these cells. So we decided to start to study adhesion in our model, and for that we used an Fmatrix microarray previously performed in the lab, comparing A375M2s versus parental cells or M2s where the contractility was lower, and we analyzed for collagen 1 receptors. And we found that amoeboid cells had a downregulation in DDR1. Moreover, DDR1 nicely correlated with the adhesion capacity of our panel of cell lines, and inversely correlated with the activation of the AMPK pathway activation and with the amoeboidness of these cells. So we further analyzed DDR1 in our model, and for that we knocked down DDR1 in elongated mesenchymal cells, which, as expected, decreased their adhesion in collagen matrices, but also induced this amoeboid phenotype, cell rounding, blebbing of the cells, and resulted in an increase in the invasive capacity, but also modified the mitochondrial network. So mitochondria became more fragmented, and these cells were less engaged in oxidative phosphorylation in 3D. Moreover, loss of DDR1 increased phospho-AMPK levels associated with an increase in the phospho-MYPT levels, and this transition to this amoeboid phenotype that was prevented when we treated cells with an AMPK inhibitor. So finally, we wanted to corroborate our data, to validate our data using in vivo models. So we used mouse models comparing tumors from, and when we compared tumors from rounded amoeboid cells to tumors from their elongated counterparts, we found that those showed lower levels of DDR1 and mitochondrial fusion proteins, and while they showed higher levels of phospho-AMPK and phospho-MLC2. And here you can see the quantifications. And we found something similar when we used a human melanoma tissue microarray, in which we compare the tumor bodies and the invasive fronts in each patient. And because, as Vicky told you before, we know that in the invasive front, there is an enrichment in rounded amoeboid cells. So we found that in the invasive fronts of these primary tumors, there was a decrease in the expression of DDR1 and the mitochondrial fusion proteins, at the same time that there was an increase in the levels of phosphorylated AMPK. The localization of the rounded amoeboid cells at the tumor edge facilitates their invasion and dissemination into secondary sites. And we know that rounded amoeboid cells are very good at colonizing the lung. So we performed the lung colonization assay using A375M2 cells, and we found that if we inhibit AMPK signaling, we are able to decrease their invasive capacity, their colonization capacity. And finally, we know that tumor edges at the metastatic site resemble that of the primary tumors, and indeed, we found the same results as in metastatic lesions as in primary tumors. So there was a decrease in DDR1, a decrease in mitochondrial fusion proteins, and an increase in the phosphorylation of AMPK at the invasive front office metastasis. So here I just summarize what I showed you today. And as a take-home message, I would say that mitochondrial dynamics control plasticity of cell migration. And we suggest that AMPK acts as a key mechanometabolic sensor at the crossroads between cellular energetics and the remodeling of the actomyosin cytoskeleton. So finally, I just want to thank all the people that contributed to this work and all the members in Vicky's lab and our funding bodies. And all of you for your attention, and I'll be very happy to take questions.
Hey, thanks for that. Sorry, I couldn't find my mute button even after a year here. So one thing that I was wondering is about cause and effect, which of course is always incredibly hard to map out. But you showed all this really beautiful data where you're sort of starting with these cell types that have different morphologies, right? So you're sort of, they have at baseline, they're either mesenchymal or amoeboid, and then all the differences between them. Do you have a sense of, or a guess maybe of in vivo, like what's driving what? Is it these metabolic changes that are sort of going to push the cells into maybe one phenotype or another, and then they behave differently? Or is it cells that behave differently or look differently have the different phenotypes? I mean, do you have any sense of that? I know that's probably a hard and probably ignorant question, but.
Yeah, I think it's hard to say that, but I mean, these cells, the ones that are around the amoeboid, they are always very highly contractile, both in vitro and in vivo. And they always adopt the same morphology. Yes, what is the chicken, which was the chicken or the egg? It's always difficult to say. Yeah. Yeah,
I know it's, but sometimes we have at least like a favorite, hypothesis for how it might go. But Adam, yeah.
Yeah. So Isabel Fabregat in the chat asks, are amoeboid cells more dependent on glycolysis for energy demands, more sensitive to glucose depletion?
So we haven't studied glycolysis at all because we didn't find any difference between their engagement in glycolysis and the different modes of migration. While we found it with mitochondrial respiration, we really focused on mitochondrial metabolism. So I don't know.
Okay, great. Sorry, I'm losing track in the chat here. From Patricia Sancho, do you see any differences on DRP1 or any other fission-related protein expression?
So we focus mainly in mitochondrial fusion, but and I also checked DRP1, and I didn't see any differences in the expression levels. I know it can be phosphorylated. I haven't checked if there are differences in the phosphorylation levels of DRP1. And I suspect that there might be differences in MFF, as that it's another mitochondrial fusion protein, but I haven't checked.
Great, thanks.
Alan R says compound C has many important effects beyond inhibition of AMP kinase, like inhibition of cathepsins and mTOR. Have you been able to confirm AMP kinase involvement by any other method?
Yes, I'm totally aware of the limitations of compound C. Because of that, we tried to corroborate always with inhibition and with the activation. So try to find both effects, so the reverse effect, because yes, I mean, we know that it's not the best compound, but we don't know any other way with a drug to inhibit AMPK. any suggestion is very welcome.
Yeah, Alan, feel free to jump in and give some suggestions there. So the next question from Ankita Jha is, do you think there are any changes in ECM microenvironment around the amoeboid cells that would influence the DDR1 signaling?
I don't really know, because we are not modifying the extracellular matrix in any of these studies that we are doing. So it might be, but I don't know. Yeah.
Great. Might be interesting to look at in the future.
Yeah.
From Stephen Thorpe, lovely talk, Eva. How do you think matrix organization and composition impacts amoeboid versus mesenchymal migration, especially in light of your DDR1 results?
Yeah, that's something that we want to study, but we haven't done it yet. So far we've only used collagen 1 as a matrix and in one single concentration. But now we would like to start playing with different concentrations of collagen, as well as with different components such as hyaluronic acid or other chemicals that we could add in the extracellular matrix, but we haven't done anything yet.
Okay, great. We do have a number more questions for you. Do you mind staying on a bit? I know we've gone past the hour, but are you okay to keep answering?
No worries, it's fine.
Great. So from Anantha, do we know whether mitochondrial fragmenting drives AMPK by triggering energy stress? Since component C is a non-specific inhibitor of AMPK, have you taken an alternate approach to validate the role of AMPK phosphorylation?
Yeah, well, I think the second part is what I said before. In terms of the first question, yes, I would say that it's, so we haven't measured the ATP or AMP after altering mitochondrial fusion, but yes, so my guess is that it would be through the alteration of AMP and ATP levels what triggers this activation of AMPK.
Okay, wonderful, thanks.
From Anna Paspera, phosphorylation of threonine 696 and 853 of human MYPT induce PPTA's inactivation, while phosphorylation of serine 472 induce binding to 1433. Did you look at phosphomyosin light chain and MYPT inactivation sites in AMPK inactivation? Do you think it's an interaction sequesters MYPT?
So yes, so we have checked PMLC and it gets modified. So when we induce this transition, so when there is an increase in the serine 472, we get an increase in phosphorylated MLC, and the other way around when we inhibit the phosphorylation of MYPT. And yes, we also checked the other two phosphorylations. They have not been described as AMPK substrates, at least not that I'm aware. But we think that when cells get contractile, there is also always a feedback loop that keeps that helps to maintain this high contractility status, and therefore, at longer times, we also see an increase in these two phosphorylation sites that very likely are downstream of ROC. That these are actually the canonical sites for ROC. And the way that it's been described, that this serine 472 impacts myosin activity is actually, yes, as Anna rightly points, through this. So when it gets phosphorylated, it gets bound to this 1433 protein, and this impairs the binding to myosin. So the phosphatase cannot do its activity, let's say.
Okay, great. We have another question from Agata Naiga. Are the mitochondrial changes just associated with the cancer cells going into the amoeboid mode, or would that happen to any cells that are not adhered to matrix, or also non-malignant cells?
We haven't checked that. I think it's a very interesting question. There are some cells, some cells in the tumor microenvironment, so some immune cells that adopt this rounded amoeboid phenotype to migrate. So it would be great to study on them whether we see the same phenomena or not. but we haven't done anything in that respect yet.
Alan followed up from earlier saying that he had published optogenetic AMPK inhibitor that arrests mitochondrial motility, and he's happy to send it.
That's great.
And then various thanks from people. I think the last one, so Sabag says mitochondrial fusion or fission oxygen reactive species. Do you think it could involve AMPK phosphorylation?
Yes, it might be. We haven't checked anything in that regard, but yes, of course, when you affect mitochondrial morphology and especially activity, you might be affecting as well ROS, so that can, yes, have an impact, but we haven't done anything in that respect yet.
It looks like just a follow-up to that, upstream of AMPK kinases like CAMK2, LKB1. Do you have a favorite?
Well, I think it's more like the levels of AMP and ATP what activates AMPK in our case, rather than CAMK2 or LKB1. LKB1 has been linked to AMPK activation through tension, let's say. So when you apply force to the cells, LKB1 recruits AMPK and it activates it. But in our case, I think it's more the energy levels would activate AMPK.
All right, I think that is all of the questions. So if anyone wants to pop in with a last minute question now, feel free. But otherwise, we just want to thank both Vicky and Eva for your wonderful, wonderful talks. Thank you so much for doing this. And all right, I'll end the YouTube live stream and we'll see you all next week.